Okay, it's 1 o'clock. So, hello everyone. Thank you for joining us today. I'm Nicole Cortiero. I'm the Assistant Director for the Institute of Cannabis Research and will be moderating today's webinar. Before we get started, I'd like to discuss a few housekeeping items. Today's presentation will be followed by a short question and answer period. To ask a question, please do so using the Q&A feature. We will address as many questions as time allows until 2 p.m. For any technical issues, please use the chat function. Both functions can be found in the toolbar at the bottom of your Zoom screen. At this time, I would like to introduce Dr. Chad Kinney, Director of the Institute for Cannabis Research, hosted at Colorado State University, Pueblo. Dr. Kinney?
Thank you, Nicole. I'd also like to add my welcome to those participating in the webinar today. For those that may be new to our monthly webinar series, I'd like to take this opportunity to tell you a little bit about the Institute of Cannabis Research. The mission of the ICR has really two main components. The first is to support and conduct cannabis-related research, and the second is to facilitate the dissemination of the results of cannabis-related research. This webinar series is one way that we can support the dissemination of cannabis research. Another way we do this is by hosting an annual cannabis research conference. In fact, the Cannabis Research Conference 2021 just launched the call for abstracts earlier this week. And if you're interested in learning more about the conference, I invite you to visit our website. I'm now pleased to introduce the presenter for the webinar today, Dr. Ashley Brooks-Russell. Dr. Brooks-Russell is a faculty member at the University of Colorado Anschutz Medical Campus. Dr. Brooks-Russell is the director of the Program for Injury Prevention, Education, and Research Program at the Colorado School of Public Health. She completed her Ph.D. in Health Behavior at the University of North Carolina at Chapel Hills and a postdoctoral fellowship at the National Institute of Child Health and Human Development. Dr. Brooks-Russell directs the Healthy Kids Colorado Survey and the Smart Source Survey. Her recent research has focused on cannabis use and the association of policy changes with changes in cannabis use and related outcomes. She is also currently investigating cannabis-impaired driving. And as you see from the cover slide, Dr. Brooks-Russell's presentation today is titled Cannabis-Impaired Driving and Questions About Tolerance. I will now turn things over to Dr. Ashley Brooks-Russell. Thank you.
Thanks so much. Thanks so much again for inviting me. I'm excited to be here today and look forward to the discussion. I have no conflicts of interest to disclose. And I'd like to start off with perhaps this obligatory map that many cannabis talks start with. And as I was updating these slides, you know, the map keeps getting greener. And I think I bring this up to just say that I feel our obligation in this field is growing to investigate the public health consequences of legalized cannabis as we see this legalization trend spread across the country. So I think this only will continue to increase in importance and relevance as we see these policy changes happen across our country and internationally as well. This next map I want to show you may be less familiar than the first map, and this is a map of state laws about impaired driving as it relates to cannabis. Many states, those in gray, don't have a specific legal limit in the blood related to cannabis impaired driving, but the ones with color do. And they vary from some states with zero tolerance for active THC or any of the metabolites, some that have set a threshold for active THC, and those range usually from 1 to 5 nanograms per milliliter, and then some states with per se laws over, with a threshold similarly from like 1 to 5 nanograms. In Colorado is similar to that, but we have a permissible inference law at 5 nanograms per milliliter. So these states have decided to set a level in the blood in the statute related to impairment. And so this talk is particularly relevant for these contexts in which we are looking to the blood to provide some indication of impairment. And unfortunately, our blood levels, the blood levels aren't particularly well suited to address this question of impairment. I want to take a step back though and provide some overall context about what's going on with cannabis use in this country broadly and nationally and in Colorado. So what we see from the most recent National Survey on Drug Use and Health is an increase in cannabis use among adults nationally. And this slide happens to be for adults 26 and up, but the same trend is occurring with young adults 18 to 26. We're not seeing it with youth, but among all ages of adults, we see this increase over time. And it starts before the years shown here. This is 2016 to 2019. It actually goes back prior to this. And we're seeing a significant increase year over year in adult use nationally. And then if you look at Colorado in particular, the context in this state as compared to the nation, this slide shows two different surveillance systems, the National Survey on Drug Use and Health, otherwise known as NSDA, and the Behavioral Risk Factor Surveillance System, some people call it BRFIS. Both these surveillance systems are plotted here. Colorado is in purple and blue, depending on which of those you look at, and the US is orange. So you can see that Colorado has been above the nation for nearly the entire time shown on this slide, 2006 onward. But the trend nationally and in Colorado is similar, is this kind of slow and steady rise in cannabis use. And the reason that I think it's worth starting with this this epidemiology is what I would propose to you is that impaired driving is likely going to increase just by a feature of the increasing prevalence over time, sort of unrelated to changes in laws and policies or that part aside. As we see a change in the prevalence of use, we're going to see a change in impaired driving. And so it's important to keep that sort of societal context in mind as we look at this issue. But now let's look more carefully and specifically about what we know about what's happening with cannabis impaired driving nationally and the epidemiology of that. We have several sources of information about this, and I like to look across these to triangulate our information on what we know about impaired driving and the prevalence. And we can look at our self-reported data, so our surveillance systems like the two I just showed you. There's also a unique roadside sampling study that's done periodically, and then we have crash data sets that can provide some information about this. So first, looking at the self-reported survey data, again, going back to the National Survey on Drug Use and Health, which provides national data on this. This slide is a plot of young adults, so 16 to 20 and 21 to 25, really our highest risk age groups here. The prevalence of using alcohol alone and then driving, marijuana alone, and then alcohol and marijuana combined. Now the top two lines here, the highest prevalence lines that show a pretty dramatic decrease over time, those are the alcohol alone lines. So I think although I haven't seen this slide updated, so I recognize it's a couple years old, I like to include this slide because I think it helps put the prevalence of marijuana impaired driving in context of alcohol, and we can see what's happening. We're seeing declines in alcohol impaired driving over time as a society. That's the secular trend that's happening. And marijuana alone or in combination with alcohol are those lower four lines that are kind of bunched together at the bottom there. But if we look at the most recent National Survey on Drug Use and Health results, this is the prevalence of, by age, of using marijuana and then driving and other illicit drugs. Those are the light blue or purple bars there. So focusing on the dark purple, we can see that the really high risk age groups for this impaired driving behavior is that 21 to 25-year-olds, followed by the 16 to 20 and 26 to 34-year-olds. And you can note there, the overall prevalence of this behavior is about 4.5%, 5%, which is consistent with about where we were or about where we've been for the entire time period on this slide, but it really varies quite a bit by age. So this is, predominantly a young adult, teenage, young adult, early adult issue that we have here. So now I want to talk about what we know from the roadside sampling information that we have. And if you haven't heard of this, this is, I think, such an interesting study design, especially for those of you who might worry about potential bias or recall bias or social desirability bias with survey data. We have the National Roadside Survey, which is when they set up research sites across the country and invite people to give a blood sample at random, and we can get this objective measure of what's actually happening on our roadways. It's not done, it's done kind of inconsistently, and we're pretty due for another data collection because the last time it was done in 2013, 2014, and prior to that, 2007. So I try to find news reports of when this would happen again, but I have to think it's sometime soon. But what we know from this study, actually looking at biological samples from drivers, real drivers on the road, is when we look at the highest risk time period, which is weekend nighttime driving. This is when you would expect the most people on the road who might be under the influence of either alcohol or some drug. What we see from 2007 to 2013, 2014, is we see this increase in any illegal drug being detected in a driver's blood, and an increase specifically in THC being detected. And then during the same time period, we see a decline in any blood alcohol level. And if you look at just 0.08, you know, the typical per se threshold, which is pretty uncommon, a pretty low prevalence there, a decline in that as well. So again, this sort of just reinforces this trend that we might see that impaired driving could continue to grow as an issue in the US, and perhaps disproportionately given the prevalence of use as compared to alcohol. What about crash data sets? We have in this country a surveillance system of crashes, fatal crashes called FARS, which is the Fatality Analysis Reporting System. And ideally, what would happen with this surveillance system is anytime there's a fatal crash, all drivers involved, whether they were fatally injured or not, would be tested for alcohol or drugs. And that would really give us a great tool to understand the contribution of alcohol and drugs in fatal crashes. Unfortunately, that surveillance system has a number of limitations, but nonetheless, researchers try to leverage what we do have to bring to bear some evidence about the role of drugs and alcohol in crashes. And this is a slide, this is just kind of an example of how these data are being used. This is Camner, that first citation in the bottom there. And what they did is look at all fatal crashes, so using like an ecological design, looking how policy changes might be related to any fatal crash, not necessarily drug-involved fatal crashes. And but researchers also have used FARS to look at the contribution of cannabis in fatal crashes, and even you can use it as sort of a proxy for the prevalence of impaired driving. But unfortunately, there's some limitations with the surveillance data. There's really large inconsistencies in states' participation in the surveillance system. So very low reporting from some states, which is likely going to reflect some bias in which fatalities are reported. Differences in how drug testing is done and how that's reported. And as it relates to cannabis, it's kind of a yes-no field that can include the presence of metabolites. So it's not a good source of really understanding if acute use may have happened before the crash. And so that's really going to put major limitations in our ability to infer the role of cannabis in a fatal crash if we don't even know when it was used before the crash. So it's not really going to give us much insight into impairment. But nonetheless, we still see a lot of research using the FARS data set and trying to make some inferences. And it is, you know, some information we can bring to bear to kind of triangulate understanding the role of cannabis in crashes and in the evidence for that. So now I want to raise the question, what about the evidence for increased risk? So what is the crash risk associated with using cannabis in driving? And certainly there's biological plausibility. We know quite a bit about the effects of cannabis and the way it affects perception and memory and attention. And these are things that we would think would be quite relevant for driving and driving performance. So some of the skills that are needed to drive, the ability to maintain the appropriate speed, appropriate distance from cars in front of you, ability to stay in the lane, and to make decisions and to drive when there's any kind of divided attention. So passenger in the car, other things that need to happen in the car, and just to maintain attention to the entire visual field, to pedestrians or oncoming cars. So from what we know about cannabis's effects, we would be concerned that it's going to impair driving performance, certainly. And so we look for evidence of this. The first place that we see a number of studies is from crash studies. So this isn't, necessarily with FARS data, but typically other case control studies where it is possible to determine or know if cannabis was used acutely. So studies where there has been blood testing, looking for active THC, or even the possibility to interview drivers in the case of crashes with injuries, not fatalities. And from these studies, and there have been a number now, we're starting to see this evidence come up from multiple reviews that have been done of somewhere between, somewhere above 1, something like a 1.2 odds ratio, all the way close to 2, maybe like a 1.8 odds ratio. So we're really seeing this evidence accumulate somewhere in this range, and it's seeming like a consistent. And just to give an example of this is one of the most recent reviews here from 2016, a meta-analysis of these crash studies that have occurred, you can see largely internationally, in fact, not all of them in the US, and over a long period of time. And what you can see is kind of that range of odds ratios. So if you're not familiar with this sort of forest plot from meta-analysis, that dotted line represents no relationships. So you see some studies going below that, some crossing the line. And then the pooled estimate there at the bottom I circled is 1.36. So again, right in that range of somewhere between 1.2, 1.8, this one, 1.4 here. So we are seeing from these studies evidence for increased risk. It's worth pointing out that as compared to the effect of alcohol on driving, this is much lower. If you could look at an effect from, let's say, a blood alcohol level of 0.08 or even 0.05, you'd probably see an odds ratio closer to something like 3 or 4. So, quite a bit larger, but nonetheless, the evidence is suggesting this increased risk. And then we also have experimental studies, and these range from studies that have some relation to driving, although maybe not directly, such as like desktop tasks on a computer, all the way to driving simulator studies in a driving simulator, and then even on-road studies, much of those being conducted internationally. And the nice thing, of course, about experimental studies is this ability to infer causation, because you can observe use, you can measure the time from use to driving, and just so much more control and the ability to infer causation here. So from this body of evidence, we see meaningful increases in impairment. Some of the metrics that we've seen include decrease in speed and increase in SDLP, which stands for standard deviation of lateral placement. And this is a measure that's really commonly used in the simulator driving world to measure driving performance, and it's basically a measure of ability to stay in the lane, or weaving, or lateral control. And the downside of these studies, for all the upsides of the experiments, the downside is it's not really easy to measure crashes, and that's really the outcome we're interested in, because that's the outcome that results in injury and death. And so we have to take a step back and get something a little more distal from a crash, and that's driving performance. So we're looking at things that we know can be associated with crashes, but typically crash itself is not an outcome, because especially with the on-road studies, that's definitely something to avoid. So that's why it's really helpful to triangulate from the epidemiological crash evidence and from these experimental studies and understand impairment. And I just want to give an example of what a study would look like that would consider SDLP, or standard deviation of lane position. This is a very recent randomized control trial that came out in JAMA last year from the Netherlands. And so this was a double blind within participant design. They had 26 participants who were occasional users of cannabis. And they measured SDLP, which is on the y-axis there in centimeters, at 40 to 100 minutes after use. And this was in an on-road driving test. And I have here some notes about the doses of THC, THC CBD, and CBD only. And one of the aims of this paper was to look at CBD only, which is not central to this talk. But what you can see here is these somewhat subtle increases in weaving that happen. The THC CBD strain there is the gray circles, and you can see that increase compared to the placebo. And, you know, this body of literature, this is just one example, but these simulator studies and on-road studies that have looked at SDLP, they've really been consistent in finding that that's one of the most sensitive measures to driving performance for cannabis. And so it's very commonly used as an outcome measure. So then we get the question, you know, what about people who use daily versus occasionally? What I didn't mention about the simulator research is, by and large, it has been conducted with occasional users. So unfortunately, we have pretty limited evidence about people who use daily. It's really only starting to get some attention more recently, and we're starting to see some studies emerge with participants with a broad range of use histories. And so that's a real gap in the literature, is recruiting participants with a wide variety of use histories. So what we know is that it is possible to develop tolerance to cannabis, and the detail is, the devil's in the detail about what can you develop tolerance to. I've included some citations for some systematic reviews here at the bottom that look at a wide range of domains. And what they see is strong evidence for developing tolerance in some domains, such as verbal learning, memory, attention, and, you know, just cognitive function. And in some of those domains, we see pretty consistent evidence of the ability to develop tolerance. Psychomotor function, which is most relevant for driving, is another area that has been looked at, although a little less often. And there is some evidence of the ability to develop tolerance to that. It's not clear to what extent, if it's partial tolerance or full tolerance. And certainly questions about the persistence of that, or, you know, if you cease use, how quickly things return to normal. You know, some of those questions aren't answered yet. So this is an area where I think there should be more research in terms of the capacity to develop tolerance, how quickly it comes on, how quickly it goes away after use is ceased, and understanding how this could relate to driving performance. And this is very critically important. And it relates back to the policy map, and I'll make that connection here in a minute. But just to summarize about the evidence for risk, of course, it's biologically plausible that cannabis would impair driving. There's evidence from crash risk, quite a bit now, that's accumulating with this odds ratio of somewhere around 1.5, and then evidence from experimental studies that cannabis acutely impairs driving performance, although the effects are smaller than alcohol as it relates to crash risk. And here's another exemplary slide, and I just realized I left off the citation here, but this is a study, Boskers is the first author, and I can get you the citation if you're interested. But this is just one of the few examples out there of looking directly at this question of tolerance, and that's why I just use this as an example. So in this study, this was a double-blind, placebo-controlled RCT, and they had 12 occasional and 12 heavy, or I would call it, you know, daily cannabis users. And they did an on-road study. So people actually drove on the road, and they looked at SDLP. Again, that's shown on the y-axis there. And they had 2 doses of dronabinol, 10 and 20 milligrams. And so you can see larger effects with the higher dose, and then larger effects for occasional users versus heavy users. And some other studies, not this studies, they looked at blood alcohol content. And so those lines of that effects are shown as the bold line is 0.5 BAC, so equivalent to a blood alcohol level of 0.05 as we'd think of it. And so you can kind of see the effect size of these doses of dronabinol as it relates perhaps to alcohol. And so this is some of the evidence that we're starting to see that heavy users or frequent users may not see the same decrements in driving performance as occasional users, although, as I said, this body of literature is pretty limited. So what do we do with this information as it relates to the real world? What are the policy implications, and what does it mean for roadside testing? This audience might be familiar with graphs like these. This is really the critical issue here. When we think about states that have set either zero tolerance or 5 nanogram per se thresholds or something of that sort, it's important to understand metabolism of THC in the blood and how that could impact assessing blood levels in a roadside context. So this is a plot from data from Marilyn Hustis, but it's mostly just a representation of the fact that THC spikes very quickly after use, quicker than alcohol, because alcohol is absorbed through the stomach, and THC, more quickly as it's smoked. It peaks very quickly and falls off very quickly. And in this figure, you're looking about approximately an hour after use. Using these data, these people would be below the 5 nanogram permissible inference limit in Colorado. And what we know in the real world is 1 hour, if there is a crash that involves an injury or fatality, it's unlikely that a blood draw is going to happen an hour after someone smokes. This, in fact, could be the time in which the event occurs, and the blood draw could be hours later than that, 2 or 3 hours. So there's some real practical challenges here of drawing blood and using that as evidence, given this metabolism curve. And the issue is although the blood level is falling, impairment is stable or potentially even rising. So this is a plot of the counterhysteresis curve that shows this relationship, and it's fairly complicated to look at, and for me, kind of, it's hard to even accept that this is how things work since it's so different from alcohol. But basically, this is a plot of what the blood THC levels look like, and then what the drug effect is happening. So if you start at the 0-0 part and then follow to the right, the THC levels are rising. This is THC across the bottom here, and time is happening. So as you get to 0.03 hours, 0.075 hours, and then 0.1 hours, during this time period, the THC levels are rising. And that reflects this sharp upward line here at right after smoking. And at the same time, the drug effect score is rising. Now at 0.1, 0.2, 0.35, 0.75 hours, we see the THC levels decline, and that reflects this sharp drop off in this figure. And yet during this time period, 0.1 to 0.75 hours, the drug effect is stable around this score of about 30. So what we see is blood levels falling while impairment is stable, and then impairment starts to decline 0.75, 1.5 hours, 2.75 hours, and then 12 hours. So there's not, the takeaway here is there's not a linear relationship between what's in the blood and the experience of the person who has used and their ability to drive safely. So blood levels peak before impairment peaks. They're not correlated. Metabolism's not linear. So a lot of individual differences in metabolism as well. And so what this means from a policy perspective and efforts to use blood or saliva or breath or any kind of marker like that, these markers are not going to identify impairment. They may identify acute use, recent use, but they may not identify impairment. There may be over-identification, given the tolerance that could develop, there may be over-identification of daily users as impaired and under-identification of occasional users as impaired. So kind of a dual challenge there with these blood levels. So what do we do about that? We want to be able to determine who's impaired or not. We want to prevent impaired driving on our roadways. So we need other ways to determine what is unsafe driving and prevent that. So that leads me to talk about our recent and current ongoing research. And I want to start by talking about some recently completed research. And I want to just acknowledge the large team that it took to pull this off. My co-PI, Michael Kosnet, and co-investigators from Iowa, Anschutz, Colorado State, internationally actually with Drivable in Canada, Ocular Data Systems, Denver Health, Understanding Legal Marijuana. So as you can see, a large team here, and a special thank you to our partner collaborator, Rocky Mountain Poison and Drug Safety at Denver Health. This work was funded by the Colorado Department of Public Health and Environment. And our primary goal with this study was to compare driving performance in people who used occasionally and daily before and after smoking cannabis. So this was within subjects design, and the focus was really on this question of tolerance. And our main outcome was a driving simulator. This is our actual driving simulator. And we also did some other assessments. I'm going to focus on the simulator for this talk. And we recruited 3 types of participants: people who use daily, daily or near daily; people who used occasionally, which was 1 to 2 times a week, no more, no less; and then people who did not use. And we asked everyone to be abstinent overnight so that we could get a sober baseline. And this was an observational study, so we set up the appointments at a time when a person said they would typically use. So for a daily user, that would typically be in the morning. For an occasional user, it may not have been in the morning, it may have been in the afternoon. And then we did all our baseline assessments. We did a blood draw, simulator drive, a number of other measures. And then we invited the people in the daily and occasional use group to smoke for up to 15 minutes. They brought their own product, flower product, typical of what's sold in the state, 15 to 25% THC content with low CBD content. And then we did all of our post assessments after that. So we could look at change within person from pre to post. And we had them drive in our driving simulator. This was a mini sim that we purchased from the University of Iowa. And some of the outcomes we looked at was SDLP consistent with the other literature. So ability to stay in the lane, speed, reaction time, mostly measured by braking, and collision avoidance. I want to share this slide. This is, as I mentioned, we took blood samples before and after using, and this is a plot of what our pre-consumption THC blood levels were. And as you can see, the non-users had none, the occasional users had 0 or undetectable or below the limit of detection. And then the daily users, quite a range here. And the scale here is from 0 to 25. And I point this out because if you think back to the policy slide about legal limits, this just reinforces this idea that this daily use group, some number of them have come into their appointment after being sober overnight, and their blood level is perhaps over some states' legal limit. So that just reinforces this concern about potentially over-identifying people as impaired if you only look at blood as your evidence here. And this is what we expected from other studies. We did expect our daily users to have a THC blood level, and so this is consistent with what we thought we'd see. And then just to show what the post-consumption looked like, notice the scaling is quite different here. This is 0 to 150 on the left there. So although it looks not that different from this figure, the scaling is very different. And you can see this very wide range in THC blood level post-use with the occasional users and the daily users. Now I just want to share some preliminary findings that we're seeing from this study that we recently wrapped up. We're submitting things for publication soon and still working on crunching all the numbers. But this is one of our main outcomes, as I mentioned, SDLP. This was measured in an urban drive context with relatively slower speeds between 25 and 35 miles per hour. And what we see, first of all, is some variation in how people drive. You know, just to remind you, our design is within subject, not between group. And so this kind of reinforces the importance of looking within person between group, because if you only looked between group, you can see they start at different points. But we have here our non-users as they go from pre to post, the pre being the squares and the post being the triangles. You see this non-significant but decrease in SDLP. That means they got a little bit better at staying in the road, staying in their lane from pre to post. And that's somewhat anticipated. We call that learning effects or testing effects. And it's the idea that people get a little more comfortable with the simulator over time. They get a little bit better at it, maybe like a video game, as they get accustomed to it. And so we see that that's also the reason that it's very important to include a comparison group. In an experimental design, this would be the placebo group, but in our case, with an observational design, it was a non-use group. And then we see our occasional users having this larger increase in SDLP, so more lane swerving from pre to post, in the daily users as well. I mean, our initial hypothesis is that we would see some evidence of tolerance among the daily use group and that the occasional use group would perform the worst. This isn't necessarily supportive of that. We're actually seeing some evidence of increase in lane weaving in both these groups. But our occasional use group, that's what's been more commonly studied in other studies. Studies have, in the prior literature, have deliberately excluded daily users. And so we can relate most closely to that literature. And we're seeing effect size approximately equivalent to some of those prior studies, on the order of perhaps 2 centimeters increase in SDLP from pre to post. So it's encouraging that it's consistent with what others have seen, although perhaps not entirely consistent with everything we thought we might see. So we look forward to getting these results published. And now I just want to tell you a bit about what we're going to do. We were recently funded by NIDA through an R01 to continue this line of research. And again, like to acknowledge my co-investigators in this work and our partner agency, Rocky Mountain Poison and Drug Safety, who helps us conduct this study. And so we now have, 4 to 5 more years to continue this work. And we're really excited to extend this line of questioning to look at edibles. Edibles presents a whole nother set of challenges as it relates to understanding impairment, with the onset of the drug effect being slower and lasting longer. We're also excited to look at high concentration products and dabs. In our prior work, as I mentioned, we only looked at flower products. And we also have the opportunity to assess more post-use. And so we're going to ask questions about the length of time of impairment and when impairment subsides, when people return to normal, when they might be safe to drive. Because those are the really key questions, I think, in this area of research. Is the public needs guidance on how long they should wait before they can drive, how they can know that they're safe to drive. And so I hope that we can contribute to that evidence and being able to give that public health guidance. And with that, I just want to end on a topic that I think maybe many on the seminar are familiar with just the challenges of conducting this research. And, welcome discussion on this. This is a picture of where we conducted our study in Aurora. We no longer rent this space in our leasing new space or planning to lease new space for our ongoing or future study. But you can just see this is not on campus, and there are many challenges with going through these sorts of steps logistically to be able to conduct this research. In addition to these federal regulation challenges, of course, there's challenges to cannabis itself. There's no single product. We've heard a lot over the past couple of years about the need to look at individual strains, to look at different concentration levels and combinations of CBD and THC. So with just such a heterogeneous product out there, I think this research is going to go on for decades and decades. And then, of course, the challenge of individual variability and tolerance, and just the wide range of use patterns that we see. Even in our daily use group, we saw people who use daily but not a lot versus people who use daily all day long. And similarly, in our occasional use group, we saw people who use occasionally a little bit versus occasionally, but on the days they use quite a bit of use. And so there's just kind of a wide range in how people are using cannabis and how that could potentially matter for driving. So with that, I'll wrap up, and I think we have like lots of time for questions and discussions.
Thank you so much. I'm going to start going through these questions. So first question is the rate of the annual fatal motor vehicle crashes decreased in contrast to the number of drug tests on roadside survey administered to drivers of fatal crashes increased in the cannabis survey?
Oh gosh, Nicole, I'm not sure I followed all parts of that question, but let me say, I mean, the challenge with looking at our fatal crash data, either in Colorado or nationally, is although it may be relatively easy to count how many fatalities we have, what we lack is a denominator. So like, for example, if we look at COVID in the recent pandemic, we see a huge decline in driving. And so, population changes, there could be, a growth in population in Colorado. And so you could see an increase in fatal crashes just because the population's growing. And you can see a decline just because people are driving less. And so it's pretty tricky to work with some of those data and make inferences about what's happening with as an effect of cannabis policies, which I think is what people want to do. And it's just has a lot of limitations with being able to answer those questions. So I don't know if that answered that question, and maybe
I think it was a multi-part question. So to the person who wrote that question, if you have any follow-up, please feel free to put it in the Q&A window. I'm going to try not to butcher the next one. How might route of administration influence the cannabis effect? Dronabinol studies like Bosker use oral administration.
Yeah, that's a great point. And I think definitely an area for research. I mean, what we know about smoked, either, vaped, dabbed, or joints, no matter the way that the smoking happens, the effect is very quick. And anytime it's, orally, it's going to be slower, and that it kind of plateaus and lasts longer. And so those are kind of two big differences. And they kind of, each of those need a lot of attention. I mean, we're seeing a huge rise in edible use, not only from, people who live here, but from tourists, who perhaps need some education as they arrive on what those effects look like and how long the effects last. So that's very important to look at those. I mean, certainly the thought is that if you're using a higher concentration product, you're going to have, more of a high, more impairment. Interestingly, we haven't looked at this really systematically, but we saw some evidence in our work, just anecdotally, of this phenomenon of people self-titrating. And so it's this idea that kind of no matter what you have, will use enough to get the effect that you want. So if you have a high concentrate product, you either use less of it or you inhale less deeply, versus if you have like a lower concentration product or a lower quality product, you do things in the way you inhale to get more out of it. And it's an important point because one of the limitations of our design is people self-administering. There's a lot of upsides of that. Let us avoid some paperwork at the federal level in terms of DEA and FDA approval and such. But the truth is even if you try to tell someone exactly what to smoke, they kind of will do what they'll do. So there's, some work that has used NIDA funding and the University of Mississippi cannabis, and what it looks like is, there's a predetermined amount of cannabis, and it's heated in like a vaporizer, like a volcano, and there's a paced protocol where you say, okay, breathe in this long, hold it, exhale, etc. Even with those designs, we're really trying to control the dose. There's just differences in how people do that, and so there's kind of a limited ability to really control what happens. And so to really get it dose in the purest way, oral is actually one way to do that, because you can give someone dronabinol and with a set amount and know that they swallowed it. And so that's just one of the challenges of this research, is this variability in dose that people take, even when we're trying to tightly control dose.
Thank you. The next one should be pretty straightforward. So what is the N for your study?
What is the end? I mean, well, I actually,
N.
Oh, the end. Oh, the end. Thanks.
N, like Nicole.
Yeah, I'm like, this is my career. Right, the end. Our goal was to recruit, that's a great point. Our goal was to recruit 30 of each group. So non-user, daily user, and occasional user. And we were successful in recruiting that many daily users and non-users. In fact, those groups were quite easy to recruit. I think we ended up with 30 non-users and 31 daily users. The occasional use group was quite difficult to recruit for, I think, a number of reasons. But one of them being, it's a pretty narrow slice of the cannabis using population. We had this narrow range of, consistently every week, but not more than once or twice a week. And that, it turns out, is just not how people use this product. So we ended up, I want to say, with 24 in that group. Our total N was 85. I think that adds up. It's really kind of an interesting pragmatic issue for people doing this research of those eligibility criteria. What we know from population studies is, among the people currently using cannabis, like using it with the definition is past 30 days, about half of those people are using daily or near daily. So it's a real bimodal distribution where, there's a chunk of people using infrequently, a few times a year, a few times a month, et cetera, and a big chunk of people using daily, and not that many people in between. And we really wanted we did want weekly users, because we really, for ethical and safety reasons, we wanted a group of people who were very familiar with the product, familiar with its effects. We didn't want people fainting or having kind of a bad experience in this research setting. So we did want people with quite a bit of familiarity. Otherwise, we could have gone with, almost anyone, a non-user off the street. But I think you really run the risk of some adverse outcomes there. So that wouldn't have been good.
Great, thank you. So I think we have quite a few questions in the queue, but we're probably only going to have time to address a couple of more. So did participants have simulator practice sessions before the study to minimize the practice effect?
Absolutely. We had a practice session when we had a first visit where we did consent and paperwork and just confirmed eligibility. And that was about an hour visit, and participants were, paid $20 for coming to that visit, and they could see the house, and we did do a practice drive in the simulator. And it's an interesting question, because there is this phenomenon called sim sickness, where being in a simulator makes people feel queasy, and it's like motion sickness, but it's the absence of motion sickness. So you're driving and you're expecting to feel things, and you don't feel them. You don't feel wind, you don't feel G-forces, et cetera, and that makes some people pretty uncomfortable. So we actually had to do the practice drive not only to kind of minimize some of those learning effects, but also rule out people who, potentially would have gotten sick during the study, which would have been, again, another adverse outcome.
Great. And last question, I think. So how does an effect of 2 centimeters translate to real world driving?
That is a great question, and one that have just been discussing recently as a research team. My co-principal investigator, who I think might be on this call, was just reviewing the literature to look at what's been published. And I want to recall that is about equivalent of perhaps a blood alcohol level of 0.05. So it's in that neighborhood. So it's likely a meaningful amount of impairment, even though it sounds very small. But again, the literature is, there's still a lot of work to be done, because there's a bit of a step from understanding how weaving relates to crash risk. And I think that's a gap in the field that could be addressed, is really making some of the linkages between these outcomes we've used in driving simulator research and what we know about crashes, and so that we know that we're studying things in a simulator that are going to directly point to crash prevention. So it is a meaningful amount of impairment, and if you take just a small leap of faith, it does relate to crash risk.
Okay, thank you. Do you have the ability to determine the dose in milligrams or kilograms of THC used by the occasional and chronic users?
I would say yes and no. I mean, what we did to try to understand dose as best we could is we wrote down all the information from the packaging of the product the participant brought in. So to the extent that dispensary testing is accurate, we have that information. And then we measured what they used before and after smoking it in a jewelry scale. And then we also counted the number of inhalations or hits that the participant took. And then we measure their blood, which is mostly just to say that we know they took in some drug, because we saw their blood levels change, although it's not a marker of impairment necessarily. So from that information, we have confidence. Oh, and we asked them how they felt. We asked them about their subjective experience of a drug effect, from 0 being no effect to 100 being the highest they've ever been, and they marked that on a line, and we measured their mark on the line. Visual analog scale is what we call that. So from these pieces of data, we know something about what they took. But as I was saying, with the variability, I mean, we don't know exactly. We could calculate how much THC was burnt, right? But we don't know exactly what they took on. But I would argue, as I said, even with paste studies, some of that's unknown. I mean, I think the next frontier for this is something that we're looking at is, could we take samples of what they bring in and get that independently tested? There's some state regulations relevant to that we're looking into. We might not actually have that ability. We're trying to understand that, and certainly changes in state law and federal law could really help move this forward, and we could independently assess exactly the product they brought in to the study.
Great. So this is actually the last one, but thanks, Ashley, for the update about your NIDA grant. Congratulations to you and your team. Just one clarifying question. Can you please remind us of what the time was for the subjects between smoking and doing the driving simulator?
Right. So they had 15 minutes to smoke, and then we gave them a couple minutes just to, rest. And then the blood draw happened half an hour after the start of smoking, and the driving simulated started right after that. So the starter driving was approximately 30 to 40 minutes after the start of smoking, and approximately 15 to 25 minutes after the end of smoking. So it's pretty close. Our goal was to measure blood at the highest or around the highest it could be. So we tried to get that blood draw pretty quickly after the end of smoking. And again, that might not be relevant for roadside testing, given that blood draws happen so much later. But for our purposes, we were trying to get near what the peak THC could be.
Great, thank you. And if folks had additional questions that we weren't able to get to today, is it acceptable for them to reach out to you?
Yeah, that'd be great.
Okay, great. Well, thank you so much for this great presentation, Dr. Brooks Russell. We really appreciate your time. For the attendees, please note that the full webinar will be available on the ICR website in just a few days. Our next webinar in the series will be given by Dr. Sarah Jane Ward on phytocannabinoids, terpenes, and neuropathic pain. This concludes today's webinar. Thanks, everyone, and have a great day. Thank you.